We're going to move over now and do X-linked crosses, and it gets a little bit more complex here because this is the different combinations of females we might have. And I've just shortened here. I didn't put a dominance letter to indicate dominance. I put an H if that indicates that the person has a mutation. I did not put an H for the dominant just to save some space. So the three different combinations for females would be a homozygous dominance, a heterozygous or a carrier, or someone that's affected. This would be homozygous recessive. For males, they have an X and a Y chromosome. That X could be normal, or that X could carry a mutation. In this case, I put the H for hemophilia. So our different possible Punnett squares are these 6 possible matings, where we have a normal female and a normal male, a carrier female and a normal male, or an affected female and a normal male. Our 3 other crosses are a normal female and an affected male, a carrier female and an affected male, and an affected female and affected male. So those are our 6 Punnett squares, and those are lined up down here. Let's start on this one. So this one's going to be the easiest one to start with because it's a normal female and a normal male. So we've got 2 females offsprings and 2 male offsprings. In this case, we've got a normal female and a carrier female. Males, we'd have a normal male and an affected male. We'll go through these again. In this case, we've got an XHX. In this case, we've got an XHX. And again, I just put the XH first. It doesn't really matter which order. Here we've got an XH and a Y. And again, another XH and a Y. Let's finish our Punnett squares first. Here we've got an X and an XH. Here we've got another X and an XH. We've got an X and a Y and an X and a Y. An X and an XH. An XH and an XH, an X and a Y, and an XH and a Y. XH, 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 XHY, XHY. Let's go through the genotypic ratios. We've got 2 XX and 2 XY. In this case, we're going to have 2 normal females, which would be XX, and 2 normal males. This symbol right here indicates a female, and this symbol indicates a male. In this box, or this Punnett square, our genotypic ratio will be 1 XX, 1 XHX, 1 XY, and 1 XHY. So if a carrier female marries a normal male, you could have 4 different genetic genotypes, which means you could also have 4 different phenotypes. You would have a normal female, a carrier female, a normal male, and an affected male. In this case, we've got 2 XHX and 2 XHY. It's going to give us 2 carrier females and 2 affected males. XHY, again, he doesn't have another copy of the gene to make up for the mutation, so he's going to be affected. Down here we have 2 XHX and we have 2 XY. The XHX is going to be a carrier female. The XY would be a normal male. Here we've got 4 different genotypes. We've got an XHX, an XHXH, an XY, and an XHY. That's going to give us 1 carrier female, 1 affected female, 1 normal male, and 1 affected male. And our last cross is going to be fairly simple: 2 XHX, H, and 2 XHY. We're going to have 2 affected females and 2 affected males. Okay, so let's broaden out and look at this one more time. Basically, what I've taken you through is some really basic genetics. Took you through mitosis and meiosis so that we'd cover both types of cell divisions, and then went into much more detail in meiosis. The process of meiosis is how you create sperm and egg, and what we try to focus on is when you divide up sperm and egg, what you're dividing up is genes, and those genes code for proteins. Now and again, those proteins will code for singular traits, and so we care how those genes get separated, because we care about how those traits are expressed. Basic ways that those traits can be expressed: there's dominant traits, recessive traits, and X-linked. We have dominant traits when only one allele is necessary to see the trait. In our example here was Huntington's. As long as one gene is making a protein that's going to clog up neurons, then you're going to see the effect of those clogged up neurons. There's also recessive, where both alleles must be present. Our example here was cystic fibrosis, where you need 2 copies of a mutated protein in order to see the trait. Otherwise, 1 chromosome is enough to make enough protein to overcome any kind of trait. X-linked was a little bit different because females carry 2 X chromosomes, and so if there's a mutation on one of those X chromosomes, she might not have that mutation on the other X chromosome, in which case she'd be a carrier, but you wouldn't see the trait. Males do not have 2 copies of the X chromosome, so if there's a mutation on that X chromosome, it's always going to be seen because the Y chromosome does not have the genes to overcome a mutation on the X chromosome. We then went through 3 different types of crosses, all the possible crosses that were dominant, recessive, and X-linked, and we worked through all the Punnett squares and all the phenotypic and genotypic ratios. I would highly recommend that you look at these tables and practice these crosses on your own. Practice getting your own genotypic ratios from these tables and your own phenotypic ratios from these tables, if at all possible. I really appreciate your attention. Thank you.